Hello and a warm welcome to everyone joining us for today's SelectScience webinar. My name is Sam Rosoman, Associate Editor at SelectScience, and I will be moderating today's presentation. I am delighted to introduce today's speaker, Michaela Bowden, Scientific Researcher at Dana-Farber Cancer Institute. Michaela will be discussing how a new automated method for RNA sample preparation from FFPE has enabled her team to overcome the usual challenges associated with this procedure. I will shortly be handing over to Michaela, but to make sure we tailor the webinar content to you, we would like to take a short poll of 4 questions, the results of which we will discuss at the end. To answer the questions, please select each poll from the right-hand side of the screen. So, question 1. What's your biggest bottleneck in NGS sample preparation? DNA or RNA extraction, library construction, size selection, DNA cleanup steps, complementary DNA or poly-A tail synthesis, other. And the question again, what's your biggest bottleneck in NGS sample preparation? Thank you for your answers. Next, question 2. You may select more than one answer here. Select the steps below that you have automated or would consider automating in the future. DNA or RNA extraction, library construction, size selection, DNA cleanup steps, PCR or qPCR setup, normalization or pooling. And that question again, select the steps below that you have automated or would consider automating in the future. Thank you. For question 3, you may select more than one answer. What type of technology are you using for DNA or RNA extractions? Spin columns, traditional filtration methods, chemical, magnetic beads, other. And the question again, what type of technology are you using for DNA or RNA extractions? Thank you. And the last question, again, you may select more than one answer here. What are the key factors in choosing automated liquid handling system for next-generation sequencing sample preparation? Individual well pipetting to efficiently automate key steps like per well normalization and pooling. Multi-channel pipetting for high-throughput full plate processing. Pre-programmed methods. Open platform with easy-to-use software that enables individuals to create automated methods. Automation company known for good customer support. Recommended or endorsed by sequencing vendor. And that question again, what are the key factors in choosing an automated system for next-generation sequencing sample preparation? Thank you very much for your contribution. We will be looking at these results after the main presentation. Just before I hand over to Michaela, I would like to invite you to post questions at any time by clicking on the Ask button at the right of the screen. We will aim to address all questions during the Q&A session that will take place after the presentation and poll results. So without further delay, I am delighted to hand over to Michaela and want to thank her for presenting to us today. Please go ahead, Michaela.
Thank you. Well, first of all, I would like to thank Beckman Coulter for the invitation to speak and to Select Biosciences for hosting this webinar. So today I'm going to talk about an automated RNA-seq library preparation for formalin-fixed paraffin-embedded tissues, or FFPE. And that was tested in collaboration with Beckman. before beginning, I wanted to reacquaint people with Dana-Farber. So Dana-Farber's ultimate mission is twofold: to provide the best cancer treatment in the world and to continue the quest for a cure through research. Dana-Farber has a proven track record in cancer research and currently holds more than 400 National Institute of Health and National Cancer Institute grants. Dana-Farber is also a non-profit entity which relies on the generosity of the general public through fundraising initiatives such as the Pan Mass Challenge, and through donations made on behalf of corporations, foundations, and individual donors to furthering our cancer discovery mission. To give you an idea of the size and scale of the Institute, here are some basic stats. There are approximately 350,000 clinical visits facilitated each year at the Dana-Farber facility, and there are over 4,000 Dana-Farber employees, which are comprised of clinical staff, nurses, doctors, surgeons, oncologists, research staff, and administrative staff. For me personally, I'm a molecular research scientist at a center which is known as the Center for Molecular Oncologic Pathology, otherwise known as CMOP, and this is affiliated to both Dana-Farber and Brigham and Women's Hospital. CMOP is also closely affiliated with the Harvard School of Public Health, and it resides in the Department of Medical Oncology within Dana-Farber. CMOP as an organization supports collaborative pathology-based research projects that have the potential to result in applications for targeted cancer therapy with a particular focus on prostate cancer. So, as I mentioned, there is a particular focus on prostate cancer, and the clinical samples selected for this collaboration are of prostate cancer origin. The latest figures from the American Cancer Society estimate that in 2013 there will be approximately 240,000 new cases of prostate cancer diagnosed. They also project close to 30,000 deaths, which means that prostate cancer is the second leading cause of cancer death in men. There is a higher rate of incidence and mortality in African American men relative to their European American counterparts. However, as of yet, the only established risk factor for developing prostate cancer is age. PSA remains the gold standard in early detection of prostate cancer. However, the major challenge in clinical research of prostate cancer is distinguishing between indolent and aggressive types that have a tendency to reoccur. Most research activities in the field focus on better understanding this distinction. Retrospective studies utilizing archival tissue specimens that have been stored in paraffin blocks represent the best opportunity to making these distinctions, since the clinical diagnosis, prognosis, and outcome in most cases is known. However, working with FFPE tissues presents its own set of complications, which have made it difficult to tap into this valuable research resource. Okay, so what are FFPE tissues? A recent article estimated that approximately 20 million archival tissue samples are stored annually in the US as FFPE blocks. This is the standard preservation method for archiving tumors. Archival collections are stored for decades and are an invaluable tool for biomedical research. Originally, FFPE blocks were cut into sections to diagnose disease by studying the tissue morphology. It is now known that degradation does occur in these blocks and particularly affects the nucleic acid composition, which has, until very recently, prevented their use for transcriptional profiling, as documented in a recent article published in Clinical Cancer Research. However, since these are vast archives, there are vast archives of these FFPE tissues available, and large cohorts of samples are required for meaningful clinical discovery. Researchers have striven and continue to strive to derive as much meaningful molecular information as is feasible and obviously reliable from these samples. Within a research hospital setting, there are two specimen collection types associated with FFPE. Number one is a fine needle aspirate. This is less invasive to the patient. It yields a small number of cells and typically can only be utilized to generate a H&E for diagnostic purposes. The second is the core biopsy. This is a much larger tissue core punch, whereby a portion of the tissue taken is sometimes frozen immediately and another portion is fixed in formalin for preservation. It is these fixed tissues that are utilized for molecular pathology research purposes. FFPE blocks are stored at room temperature, which is of no extra cost for refrigeration or cryopreservation to the institution, and there is also an obligation to archive these samples for 25 years post-biopsy, therefore resulting in a large accumulation of these preserved blocks. In particular, prostate cancer cannot be diagnosed very well on frozen sections due to the difficulty in assessing the morphology of these heterogeneous tumors. So predominantly prostate cancer tissues are stored as blocks. As a result of, and in parallel to these clinical archives, there are large long-term voluntary initiatives such as the Physicians' Health Study, the Physicians' Health Follow-up Study, and the Swedish Watchful Waiting Cohort, whereby tissue and blood specimens and biannual medical health status questionnaires are completed by participants. In the case of prostate cancer, through these initiatives, prostate cancer-specific cohorts have been derived. Through our collaboration with Harvard School of Public Health, we have access to more than 3,000 prostate cancer FFPE specimens. Okay, so back to the factors which influence the RNA degradation rate of the biological specimen. So the upstream process of fixation has a profound impact on RNA degradation. Three scenarios are depicted in the schematic shown here. In the first instance, fresh tissue is represented. Proteins in nucleic acids are unbound and can easily be purified and extracted from the tissue, resulting in high-grade quality RNA suitable for many types of downstream applications. In the second instance, right after formalin fixation is shown. Through the process of fixation, cross-links bind the proteins in nucleic acid, which represents a more challenging specimen on which to perform purification and extraction. RNA quality should still be reasonably good. However, some of the RNA fragments will be broken. The third instance represents the effect prolonged formalin fixation has on the sample. Cross-linking is severe in this case, and purification and extraction processes only partially maintain the integrity of the RNA, resulting in highly degraded and low-yielding samples, which have limited downstream application potential. The cross-links are unable to be broken and very little of the RNA remains intact, resulting in highly fragmented and overall bias and an overall bias towards small RNA preservation. So the biggest factors impacting fixation are the thickness of the tissue specimen, the duration of the fixation process, and the volume of the formalin solution used. As well as fixation, there are additional factors that influence RNA degradation. These are the age of the tissue block, the water composition of said tissue block. There are two ways in which water can affect degradation levels. Either water is retained during the fixation process, or over time the block is stored at room temperature. It's exposed to humid ambient air, which also has an impact on the quality. Storage temperature, which is linked implicitly with humidity and air exposure, which results in slow oxygenation of the block. FFPE tissue blocks present their own unique challenge in unlocking their potential for molecular discovery. Okay, so to switch gears a little bit after that introduction to FFPEs and prostate cancer, I would like to move on to the specific goals of the project that we set out to accomplish. So the primary goal is to implement an automated method for preparation of RNA-seq libraries from archival FFPE tissues using specifically the TruSeq kit. So in order to achieve this goal, there were a number of factors that had to be looked at. We had to look at the optimal amount of RNA input to be used to generate these libraries. We wanted to look at the size of the RNA-seq library that was generated from degraded tissue and how that looked compared to fresh frozen or cell-based library prep. We also wanted to establish FFPE-specific purification methods that may be needed to employ to clean up library prep prior to sequencing. And of course, ultimately, with any new method, we wanted to ensure that an automated method would generate comparable sequencing data to a library that was prepared manually at the bench. So in order to accomplish our goal and to work through some of the parameters, we set up a structured workflow following in sequence the 9 steps shown here. So before beginning this study, we wanted to select suitable representative degraded archival FFPE tissue specimens. In-house tissue blocks were acquired, so we request them through an internal database system and were reviewed by a pathologist. And the criteria for selection were to choose similar morphological specimens with matched low Gleason score. Gleason score is the grading of the tumor, prostate cancer tumor. To choose specimens that were less critical to our overall research endeavors, since the purpose of this collaboration initially was to develop a technical method that could then be utilized on precious samples of greater importance. And what I mean by that is, obviously these blocks are of a specific size, and there's a finite amount of material available to be used. We didn't want to use blocks that were critical to any sort of molecular discovery going forward for method development. So we chose blocks that were less a priority for some of the research endeavors that we have right now. And having said that, having developed this method, we will then apply it to all of these precious samples that we have. And then a final criteria was to select tissue blocks where there was a pronounced tumor region of interest that would not be exhausted through extensive coring. So these cores were selected and processed and the RNA extracted. The RNA was then quantified and the quality was assessed to determine the level of degradation. The automated libraries, followed by optimization of the cleanup step, was undertaken first. This was followed with the manual library prep on the bench and subsequent library cleanup. The RNA-seq libraries for both were submitted to the Dana-Farber Core Service at the Center for Computational Cancer Biology. Upon receipt of the sequencing data, basic performance metrics were documented and a comparative study of the sequencing data derived from the automated and manual library prep was undertaken. Okay, so moving along to the sample selection. So we selected 3 individual specimens and secured matched tumor and normal paired FFPE blocks for each. Upon pathology review, it was determined the Gleason score for all 3 tumors was 3+4. All sample IDs have been masked to protect patient identities and will simply be referred to as prostate tumor A, benign A, prostate tumor B, benign B, and prostate tumor C, benign C in the following slides. H&Es were generated to annotate the tumor in benign ROIs, which were used to locate the area of the FFPE tissue blocks to take multiple core punches from. So the core punches taken from the FFPE blocks were processed and RNA extraction was undertaken. We used the former pure kit for all extraction procedures. The RNA was then quantified by ribogreen assay, which is a reagent-based colorimetric measurement of the RNA present in the sample. The color change associated with RNA amount was read on a Perkin-Elmer spectrometer. The RNA concentration was calculated from the standard curve generated. For the 6 samples in this set, the RNA concentration levels vary dramatically between 25 and 60 nanograms per microliter. We assessed the quality of the isolated RNA on a bioanalyzer. The figure shows the digital gel readout for 2 representative samples, in this case tumor A and benign A, relative to an RNA ladder. On the right-hand side, the resulting electropherograms are shown. These are plotted as time versus signal intensity, which can then be converted to fragment size. Typical plots of good quality RNA have a distinct peak representing 18F and 28F, and the peak ratio of which is used to derive quality of RNA. from the plot here, it is evident that both of these peaks are absent and that only small RNA fragments are present, resulting in RNA that is highly degraded. The differences in yield associated with area under the curve correspond to the concentration levels as measured by ribogreen assay. So we actually tested multiple RNA input concentrations on the bench and went as low as 100 nanograms, but we determined that 500 nanograms was necessary to generate RNA-seq libraries. We don't actually show the data here, but we thought that that was a safe RNA input amount to ensure that all samples would just be able to generate a library. Okay, so the TruSeq library prep comes with an extensive step-by-step method description. So I'm not going to get into a lengthy discussion about that today. However, for the purposes of adopting this protocol to an automated platform, the method can be loosely broken down into three main parts. These three parts correlate with stop points, and a stop point is defined where the method can be stopped and the sample can then be frozen overnight, and you can continue on with the method the next day. So step one encompasses the mRNA purification and fragmentation, and then follows up with the first and second strand cDNA synthesis. Step two revolves around construction of the cDNA library. There are multiple steps in this, which include repair and A-tailing and adapter ligation. And then step three represents amplification of the cDNA library and overall PCR cleanup. To start out, we incorporated hard stops after each step. However, now the steps are routinely run consecutively to speed up the entire process. Okay, so here we have representative plots of the cDNA libraries that were generated. So an aliquot of each of these libraries generated was run on the bioanalyzer using the DNA chip to generate these plots. And so on these plots, what you can see is the x-axis represents the size in base pairs of the library generated. The y-axis is signal intensity, which is associated with the yield of the library. In this particular example, we show tumor benign pair B. On the left-hand side, we show the preps for tumor benign that came from the automated prep, and on the right-hand side, we show their corresponding manual libraries. So the library yield is greater for the automated prep. If you look at, I've highlighted the signal intensity on the y-axis for each of the plots. So they're not all scaled to the same height, but the yield is much greater for the automated preps, and as a consequence, you can see that the area under the curve is larger, and this results in an overall size range that is larger for the automated preps relative to their manual counterparts. And again, this is done with the same amount of 500 nanograms RNA input for each of these. Okay, so we also spent a considerable amount of time determining the best approach to post-library generation cleanup, and in general, cleanup steps on the multiple steps along the way of the process. So in high-grade RNA derived from cells of fresh frozen tissue, and the amount of RNA present throughout the process is always in excess. Therefore, anomalous peaks and/or primer-dimer formations are generally minimal. With the wide-ranging variance in RNA quality derived from FFPE, reagent components and inline controls and/or adapters are typically present in excess, which results in a more pronounced challenge in terms of cleanup. Although not shown here, we tried alternative cleanup approaches using spin columns and gel band extractions to achieve our goals. However, we have had the most success with bead-based methods as described here. So for this particular study, we ultimately used this method that I'm going to describe here, which is a SPRI select fragment size selection. So initially, on the plots on the left, we're using tumor A and benign A as our paired example. And we've taken the data generated on the bioanalyzer and overlaid the pre and post cleanup steps. So on each plot, the black line corresponds to what the library looked like prior to any cleanup, and the red shows what the library looks like after we employed this bead-based two-step strategy. And on the top, we have the benign library prep is shown, and on the bottom, we have the tumor. So initially, these libraries had a peak max for the amplified cDNA at about 260 base pairs. However, in all 12 of the samples, we had a non-specific adapter amplified peak at about 120 base pairs. So that's highlighted in blue. And you can see how it corresponds to the low and high molecular markers within the trace. So an additional fragment of greater than 400 base pairs was also present in some samples. This was particularly present in the tumor samples, actually. And so we wanted to use an iterative process to see if we could remove the lower peak and the upper peak while not compromising the integrity of the library. So with this SelectScience beads, we were able to employ this two-step strategy. So once the libraries were generated, for step one, we mixed the library with a 0.7x ratio of the SelectScience bead solution to cDNA library volume, and fragments greater than the 500 base pair DNA bind to the bead. And these beads are then discarded. Then we take the remaining supernatant for step two, and we treat it with a 1.1x volume of SelectScience beads to rebind the DNA fragments between 150 and 400 base pairs. And what you can see if you look at the red traces in these, this is in the automated process, we were actually able to completely remove this low molecular band at 126 base pairs post cleanup with these beads, and the significant peak of a greater than 400 base pairs is also removed. Overall, there's some loss in the quality or in the yield of the library, but not enough. But the pros of cleaning up this library outweigh any small loss we associate with yield. So moving into actually looking at overall how the sequencing was performed and then what it actually looked like. We ran the libraries were transferred to CCCB for sequencing, and the sequencing was performed on an Illumina high-speed platform. We chose a 50 base pair single read workflow, and these multiplex sequencing was performed in such that for each lane contained 3 samples. So 50 base pair single reads were mapped using top hat and transcript abundance in FPKM units, which are fragments per kilobase of mRNA per million reads, was calculated using coupling. So we have some bioinformatics experts who handle all our sequencing data, and so they generate all this data for us. And so RNA-seq library construction and sequencing was successful in all 12 samples, with a varying alignment reads ranging from 21% to 72% relative to the total yield, as shown in the table. So the biomass samples were bundled in flow cell 1 and 2, and the manual samples in flow cell 3 and 4. The tumor C, benign C pair was the only sample pair that were run on different flow cells for each. The total number of reads generated per sample, although large, varied greatly from sample to sample, as did the percent reads that aligned to the genome. We believe these variances are mostly associated with the variance of the quality of the RNA extracted from FFPE tissues. We have started to implement new strategies around ribosomal depletion to bring more uniformity to the RNA input that goes into library generation. And we are also working on developing new algorithms to compensate or account for loss of quality that can be applied post-data generation. However, the take-home message from this initial pilot collaboration with Beckman was that libraries generated by automation look similar to those generated manually, and in some cases look superior. And in order to further explore this concordance, we set out to assess the transcript. Okay, so this is quite a busy slide, and it's dense with data. So basically what we did was we generated scatter plots from the transcript data that was derived from the sequencing. And so in this way, we were able to do a direct comparison between the methods for each individual sample. So in this study, just to remind, there was 3 tumor normal pairs, 6 samples run with 2 different methods, one automated, one manual. So in each plot here, the x-axis represents the transcript data generated from the automated library, and the y-axis represents the transcript data generated from the manual library. And the data is plotted as log to the base 2 of the SPKM value, and so they were used for this correlation. So as you can see, the reproducibility was measured by calculating the Pearson correlation coefficient from transcript abundance values. And as you can see, in most of these cases, the Pearson correlation is above 0.9 between the automated and manual prep. So you can see that there's good concordance between the 2 methods for the same sample. So we also showed what actual transcripts we use. So there's 2 data points or data information that's given: the total number of transcripts that were actually found, and then the value of those, the total number that had a greater value than 0 is also displayed. One thing we will point out, and this is mostly to do with some of the differences that we see with varying quality from FFPE tissues. In particular, you can see that tumor benign pair A, tumor benign pair B look very good, and tumor benign pair C look less. The correlation isn't as good. The Pearson coefficient is below 0.8, and in this particular case, we can correlate this directly back to not only what the RNA looked like for input, but also what the library generated looked like before we sent it for sequencing. And so it looks as though for FFPE, as we currently work, no one input value of RNA is going to be suitable for all sample types. And so, we have to come up with smarter methods about how we determine how much RNA input material to put into these, and also to understand that these blocks are finite, and we can't put in, also while trying to reduce the amount of input material that's going in overall. Okay, so there's any wide-ranging number of endogenous controls that can be employed for quantitative gene expression analysis in clinical tumor samples. And typically these are employed to correct for any sort of cellular differences or RNA quality variance you may see from sample to sample. So this particular panel is comprised of 27 housekeeping genes. Roughly half are well-known genes, and another half were ones that were determined to be potentially useful for large, high-throughput data sets, like from microarrays or from sequencing technology. And so utilizing this panel, we wanted to compare the manual prep to the biomet prep. So what you see in this graph here is it's a scatter plot of these 27 endogenous controls. And what we've done is, again, it's the SPKM values are log to the base 2 plotted manual versus biomet, and these values were calculated as the average for each sample. So for each individual sample, we took the average value for that particular housekeeping gene and plotted it relative to the average value for the robot. And so you can see that there's a concordance of 0.97, which is pretty much exceptional. And just to show what these plots look like, I pulled out 3 representative housekeeping genes and plotted the histogram with the error bars to show how the robot and manual compared. So we took a low responder like G6PD, we took a mid-range responder like OAZ1, and then we took a higher responder like GAPCH. And across the board for all 27 of the housekeeping genes we selected, we saw this great concordance between the automated data and the manual data. So this made us this is kind of like the go no go for implementing an automated method, because obviously the automated method is far quicker than doing it by hand on the bench, and it lends itself to high throughput. Okay, so just to summarize some of the findings that we had, we were able to generate these RNA-seq libraries utilizing an adapted TruSeq method from as little as 500 nanograms RNA of FFPE origin. And these automated cDNA libraries had a better yield than those prepared manually. And at the transcript level, the matched biomech and manual prep samples showed very good concordance. So just some notes on the TruSeq method. It is actually designed for high-quality RNA and works extremely robustly for RNA of cell and fresh frozen origin. And we routinely use this in-house in an automated fashion. We provide a core service for our sequencing facility in this matter, and we really like working with the TruSeq method. And so there was no real optimized protocol at the time we started this study for FFPE specimens. And one thing that is abundantly obvious from this data is that we would like to remove all ribosomal RNA to sort of have more uniformity of the sample that is input into any sort of a library prep method. And we had done some preliminary work on this, and it had shown that we probably needed too much input to begin with to actually make it work. So we're now working on strategies around how we can use ribosomally depleted RNA as an input at these lower concentrations. So going forward, it is envisioned that new coding and non-coding transcripts, as well as gene signaling networks that strongly associate with prostate cancer progression, will be identified when we shore in these methods. And we absolutely think that the biomech automated platform offers a viable high throughput alternative to any sort of traditional manual bench preparation for any type of RNA libraries for sequencing. So obviously there are a number of people to thank who were involved in this preliminary collaboration. So from the Dana-Farber Cancer Institute and the molecular technician who did a lot of the automated developmental work along with Beckman Coulter was Ashley Rossi. Our resident bioinformatics expert is Svetlana, and she generated some of the basic metrics around this, and also she's now using this data as part of bigger data sets to look at the comparison between fresh frozen and FFPE tissues to determine what we actually lose when the sample gets degraded, and is there ways we can compensate for that. And obviously my direct boss is Massimo Loda, who's the director of CMOP, and also an expert in cancer, prostate cancer. And from the Beckman Coulter end, we worked very closely with Dina Lee, who's a senior application scientist, and Sarah Simmons, who's a field application specialist, who helped develop the protocol for the system and helped with actually any sort of teething problems getting it up and running. And so with that, I will hand back over.
Thank you for that informative presentation, Michaela. We do have some questions for you, but before we look at those, let's take a look at the poll. We actually had a bit of a technical problem with poll 3, so I'm actually going to ask you all to take a look at that one again. So for question 3, you may select more than one answer. The question is, what type of technology are you using for DNA or RNA extraction? extractions. Spin columns, traditional filtration methods, chemical, magnetic beads, other. And that question again, what type of technology are you using for DNA or RNA extractions? Thank you. Okay, so if people would have a look at that and just let us know what they think, that would be great. So we will now move on to the results from the other poll questions. So for the first question, let's have a look. So we can see here that library construction is the biggest bottleneck in NGS sample preparation. So that's always interesting what other NGS users consider to be their biggest bottleneck. And then let's have a look at the results of question 2. So, yeah, it's obvious here the biggest step to automate appears to be, I think that's library construction. Okay, so like I said, we will come to question 3 at the end. We will also be looking at question 4, and we'll be doing those after the Q&A section. So we've had quite a few questions come in whilst you were speaking, Michaela. So we'll just move on to the last part of the webinar. So let's begin with this first question that we have for you. So you used 50 BP single-end reads, which is great for getting counts and looking at differential expression. What are your thoughts of using FFPE samples to look at alternative splicing or even more challenging SNP information when using a more comprehensive sequencing protocol, i.e. 2 × 100 BP?
Hi. Actually, well, we haven't actually done that, but we are in the process of, I think this, for a pilot study, we just decided to go with a straight-up 50 base pair single end. We actually, for some of the projects that we have, we use 100 base pair end reads, and we're looking, obviously we have a lot of samples, and for these sort of cohorts, we look at anything from 100 to 200 clinical specimens, and there's a huge cost associated with, the brute force end. There's a huge cost associated with running these sort of sequencing endeavors, and so usually we have to tie them to any grants that we have. So we haven't done it. We've definitely thought about it. I know it's something that our bioinformatics team are interested in, but that's as much as I can say in it right now.
Okay, thank you very much for that. So another question we've had is, do you have a feel for how the manual versus automated comparison would compare to just 2 manual preps of the same sample? I.e., is this a normal amount of variability that should be expected with FFPE samples?
Yeah, I think so, because I think from the data that we showed, we had 3 pairs of samples, and they were both run on a manual, they were all run manually, and then they were run on the robot. So we actually do have, 3 samples to compare across manual. And so we had the exact same pattern, which is that sample specimens A and B looked very good and very similar, and sample C, both manually and on the automated system, behaved the same. They didn't have the same concordance level. So when you compare, if you just take an individual method on the bench and compare it, it also stood out relative to the other 2. So I think it is a variability that's associated with FFPE samples. I mean, another reason why we were trying to use the lowest amount of RNA input we could get away with and still generate libraries. I think maybe if we'd have used, 1 microgram of FFPE tissue, that somehow you can compensate for some of these variances. But 1 microgram of tissue is just not feasible going forward with with these precious samples. Sometimes that's all that may be available, and since these studies are involved for retrospective studies over many years, we don't know 5 and 10 years in the future what you may want to use them for. So we have to be very careful about how we use them right now. So we're always driving for what can we do with the lowest amount of material that can go in. So we think there are things that you can do on the back end. We run match studies with, as I said, fresh frozen matches for some of our samples, and we're looking at developing algorithms to actually compensate somewhat for what is lost in the FFPE sample.
Okay, thank you very much. So moving on to the next question. How old were the FFPE blocks that you used? Were they roughly the same age, the same time stored post-fixation? Have you ever noticed that older blocks have a lower RNA yield?
Okay, so the blocks that we used here were from 2006. We matched them as close as we possibly could. So for all annotation of any clinical samples, we're able to derive when that sample was taken and when it was embedded. So we have all that information and we matched them as much as possible. So they are all the same age. They were all stored in the same facility, in the same room for the majority of that time. So they were all exposed to the same sort of conditions. And so the fixation, I mean, there's a standard protocol within the clinical environment that is used, and so they would have been subjected to the same protocol. So obviously maybe there is some person-to-person variance associated with that, but essentially they were all fixed with the same protocol. For older blocks with a lower yield, yes, we definitely do see this. So we have some blocks that are about 25 years old. So this is obviously run on a cohort of samples from 2006. Some of the studies we're working with now, some of the samples date back to maybe 2000, 2002, but we have an older subset of samples that go back 20 years, maybe even longer. These ones we would really like to start to work with in a sequencing capacity, but until we're very confident about how much material we can use, and what we can actually do with them, we don't want to touch them. So we definitely know that when the blocks are very old and they've been left and subjected to basically ambient temperature and air in a room, that their yield, the cross-linking should be very severe, and the yield goes down, and there's definitely less RNA there. And having said that, we do now have other projects where we're actively collecting tissues and fixing these tissues. And some of these we've started to, for other methods actually outside of sequencing, for LCM purposes, which is laser capture microdissection, we've started to store these blocks directly at 4 degrees in desiccators, and this actually staves off the aging process. So it doesn't stop it completely, but it certainly perturbs it dramatically. So these blocks maintain their integrity. They have obviously, due to fixation, there is some cross-linking. But after that, if they're maintained at 4 degrees in a controlled environment, then we're able to minimize the impact of excessive other conditions that may affect the yield. And we've actually shown that differences between blocks stored at room temperature and at 4 degrees show significant difference in yield and actually of quality. We're able to maintain some of the quality of that RNA.
Okay, thank you very much for that. So moving on to the next question. Could you estimate how many libraries can be prepped on the Biomex system per day or week?
So we would comfortably recommend a full 96 well plates as we currently stand. I am sure that Beckman would have ideas about how you would process multiple plates, but currently the way we're set up, we would be comfortable doing 96 plates over maybe a long day. It would be maybe over, I think we determined it was about an 8 or 9 hour day. So if you really wanted to, in a given week, you could do up to maybe 500.
Wow, okay, thank you. Moving on to the next one. Do you have to fragment RNA as it is already fragmented? What is the 400 BP band that you have to remove in purification?
Yeah, that's a good question. We've discovered with FFPEs, you really don't. We looked at, so for the TruSeq protocol, I'll just take it back. For the TruSeq protocol, there is optimization of the fragmentation time no fragmentation. And so if you don't frag at all, there's no difference in how the libraries are generated. So now we don't, for FFPEs, for TruSeq method, we don't fragment the samples anymore. The second band at 400 base pairs, or it's actually above 400 base pairs, it's past that point. We are not entirely clear what that band is, and I think there's some discussion in the field around, and other people have seen this double peak as well. We've since used alternative methods for generating libraries. We don't see this second peak, so in this particular instance, we decided to remove it so that we could compare it to the manual prep, because we didn't actually see that peak in some of the manual prep. And having said that, it would be interesting to be able to sequence that and see what the difference is, but we don't actually, we don't have a clear idea of what that is. We just know it's probably not representative it's not representative of our samples in our FFPEs, because really they only contain small RNAs.
Okay, thank you. And moving on to the next one. Did you do poly-A pull-down since you didn't do rRNA depletion?
Yeah, for these particular samples we did. But again, as I said, we started to moving to new sort of ways of optimizing. We actually now would, I think initially when we started to work with ribosomal depletion and different library preps, we didn't have great success in generating enough yield of a library, and then when we tried to amplify it up, we ended up amplifying anomalous peaks as well, and they became very difficult to remove. But now we've come up with a new strategy of how to do this, and so moving forward, we plan and intend to ribosomally deplete our FFPE before generating a library.
Okay, and another one that's come through. What's the advantages of using the standard TruSeq method you outlined over using the standard TruSeq prep, which is capable of using FFPE, RNA, and ribosero step?
Sorry, could you say that again? I didn't quite hear the beginning of the question, sorry.
Yeah, sure. What are the advantages of using the standard TruSeq method you outlined over using the standard TruSeq prep, which is capable of using FFPE, RNA, and a ribosero step?
So from the data that FFPE data that we've generated with these FFPEs with the TruSeq standard method, when we don't deplete they haven't been depleted for ribosomal RNA. The ribosomal component makes up close to 50% of what's actually present in that sample. When we look at what's present in a fresh frozen, the ribosomal component is a lot lower. And so we know that now that we would like to remove that ribosomal RNA and modify the prep so that even if the library is quite small, we'd rather have a clean library going forward for sequencing purposes than not. I think if that answers the question.
Okay. And the next one. Was the cDNA synthesis done manually or with automation? If automated, can you provide some details?
Yeah, the cDNA synthesis was done with automation. Some of the thermocycling steps were done offline, but there is a way in which it can be done on the system. For this particular study, we took some of them offline, but I know there's thermal heaters that can be used to do this all completely online. So we have the protocols by which to do this. Just for this particular study, we took the thermocycling options off.
Okay, thank you very much. So another one that we've got through. How did you modify the original TruSeq protocol to avoid mRNA isolation and fragmentation?
So from the original protocol, we actually, the automated method, we just brought it through that method entirely. We did follow all of those steps as documented in the original protocol. And since then, we went on and looked at ways to optimize and remove some of these steps, but we found there was very little difference in the library that was generated.
Okay, thank you very much. And another one that's come through. Have you considered double-strand specific nuclease treatment to get rid of the rRNA in your samples?
Yeah. Actually, prior to this collaboration, we were doing some bench press with FFPE and testing out different parameters in general. And we actually did look at the DSN method, and we found that it wasn't very helpful in removing the ribosomal components. So we don't use that, and we don't intend to use it going forward.
Okay, thank you very much. We've got a lot of questions coming through. Another one is, what is the yield of cDNA and the PCR cycle number used for library amplification? We send our libraries to our core service, and they run qPCR validation on them, and then they flag whether they're suitable for sequencing or not. Having said that, sorry, what was the second part of that question again? Apologies. Yeah, that's fine. Bear with me one moment. So, what is the yield of cDNA and the PCR cycle number used for library amplification?
Sorry, yeah, it's the PCR cycle number. So we use 15 rounds of PCR for our application step. We looked at using 12 cycles to avoid generating primer-dimers and some concatemerization. We looked at this. We also looked at it amplifying more and trying to compensate for any noise in the library by cleanup. But essentially, we stuck with the 15 cycles.
Okay, thank you. So this will be the last question. So what is the percentage of rRNA in your final data?
Yeah, so the percentage of ribosomal RNA in our data it varies from sample to sample, but essentially across all kind of FFPE samples that we've looked at, it hovers around the 50 to 60% mark. So it is quite pronounced in these particular sets of samples. And I think maybe just a point I'll make on that is that obviously I think the best comparison we would like to make is we would like to look at these libraries, and then we'd like to look at ribosomally depleted libraries from the same samples and compare whether there's anything to be garnered from removing it or not. We think there is, but because these samples are so degraded, we're not sure how much is to be garnered from them.
Okay, thank you very much for answering that very large number of questions. Okay. So we will now run through the results from questions 3 and 4 from the polls. So question 3, we can see here that the majority are using spin columns. Okay, and then finally for question 4, it seems that the biggest factor for choosing to automate NGS sample preparation is recommended or endorsed by a sequencing vendor. So we've got some similar results here. So yeah, they're very similar to each other. The percentages are not far off, so quite interesting thing to note there. Okay, so that's all we have time for today. Thank you very much, Michaela, for today's informative discussion and presentation.
You're welcome.
Thank you to everyone joining us online and for your interesting questions. So anyone we haven't had time to answer, we will get back to you personally. So those will be forwarded on to Michaela. I hope that everyone has found this a worthwhile session. So if you do have any more questions, please feel free to email me at editor@selectscience.net. If you would like to listen again to today's webinar or invite a friend to listen, it will be available on our website shortly. So goodbye and thank you once again.